Hello, my name is Adriana Toma, and today I'll be presenting on this study done by medicinal chemists surrounding breast cancer cell growth. Now, the title of this study was Serotonin Analogues as Inhibitors of Breast Cancer Cell Growth. It was published in ACS Medical Chemistry Letters, and as you can see, the authors are as written. Now, the study centers around serotonin, which is a molecule that's not only a neurotransmitter, but also acts within tissue, and it helps in regulating cell division, growth, and the overall health of epithelial cells, as well as lactating and involution. However, when the serotonin signaling becomes abnormal, it can contribute to breast cancer by, instead of regulating the metabolic activity of the cell, causing increasing cell proliferation and making the cell resistant. This is why they chose to study serotonin as the main molecule of this study. Now, if you look at the structure of serotonin, or as in its chemical name, 5-hydroxyltryptate-amine, or 5-HT, you can see that there's an endo-ring system made up of a benzene ring and a pearl ring fused together, which helps in stabilizing the molecule. There's also a hydroxyl group, which helps forming hydrogen bonds with the receptors, and it's really important when discussing serotonin receptors, which is integral to this study. There is also an ethylamine side chain, which is a 2-carbon chain ending in an amine group that is part of the tryptamine backbone, which is also very important to the structure of serotonin and its involvement in breast cancer. There's also the primary amine, which is often G protein coupled receptors. These are receptors that are surface level that can detect external signals like hormones, neurotransmitters, odors, and light, and then because once they receive those information, convert them into an intercellular response, causing something to happen in the cell because of external stimuli. They have 7 alpha hexacycle segments that cross the cell membrane, loops for ligand binding, and intercellular loops that interact with G proteins so they can activate them. They are the largest receptor families in humans. There's over 800 types. Now, how do these G protein coupled receptors work? First, there's a ligand. The ligand binds, so a hormone, neurotransmitter, or drug binds to the receptor, and then the receptor changes because of the binding. The receptor changes its shape, activating the G protein. This then turns the G protein on. The part of the protein that binds to the GPD drops it and then connects to a GTP. So basically, a GDP bound G-coupled protein is inactive while a GTP-bound G-alpha coupled receptor is active. Then what happens is something called downstream signaling with 2 parts of the protein free. They then separate and then go on to activate other parts of the reaction. So the main G protein coupled receptors that are in this study, which relate to serotonin, is GS, which is linked to 5-HT467, and then GI, which is linked to 5-HTI, and then GQ, which is linked to 5-HT25. So when talking about dysregulation in breast cancer and how that relates to serotonin, it starts with an increase in serotonin production that's caused by the overexpression of the late-limiting enzyme tyrosine hydrolase. And as the production of serotonin increases, there's a shift in the balance of signaling. So as some receptors increase their signaling and cause reactions, others go down. So this causes an imbalance. And because of the imbalance, instead of promoting the regulation of cell growth, serotonin turns into pro-growth transmitter, which causes increased levels of growth. So the goal of the study was to design, synthesize, and test a serotonin/tryptamine analogues to identify if they can induce apoptosis, which means to decrease the level of, or cause the cell to die. And their hypothesis was that the modifying structures they created could produce compounds that could break down cancerous cells. Now for the materials, they used a lot of different materials when it came to the cell lines and cell culture reagents, as you can see below. There's also a bunch for the chemical synthesis of the products itself, they used a bunch of different chemicals and different tools, such as drying agents, TCL plates, silica gels. Now for methodology and synthesis. They started off with the cell culture. They took breast cancer cells from ATCC and maintained them in DMEM/F12 solution, which had 10% FBS. Then these cells were used for no more than 3 months. So once they reached the 3 months expiration period, other cells were used. Now, to begin, they started with the synthesis of the serotonin analogous compounds. There was first a protection step, where the serotonin hydrochloride and potassium carbon monoxide were stirred in a water solution with CH3CN. And then BOC2O was added, and the mixture was stirred for 24 hours continuously. Then there was the extraction purification process, where the reaction was diluted and was extracted with ETOAC, and then it was washed, drained, and then concentrated to yield the N-BOC serotonin molecule that they were looking for. And then that molecule was dissolved in CH3CN with K2CO3, and then reacted with an alkyl bromine at 80 degrees Celsius for around 24 hours. Then the deep protection process took place, where an alkylated BOC intermediate was stirred with 3 molar HCl and an etoO acetate, this generating the target products as hydrochloride salts. Then these were then isolated through filtration and washing. Then they synthesized 4 and 7 alkyoxy triptamine. They started by the alkylation of hydroxynidolide with K2CO3 and nonylbromide in a refluxing acetonitrile, in the formation of a nitroethylene intermediate, which was then reduced in THF to produce a final aminoethyl derivatives as chloride salts. They then took further steps, where the cells were plated in 94 well plates and allowed to attach overnight. They were treated with various concentrations and then were measured using a CL titer aqueous 1 MTS reagent. The absorbance was as 49 nanometers and was recorded using a Victor plate reader. Cells were then using the same plates, were lyzed and analyzed for adipose using Capsi's Plo. Then the lubricants was recorded. Cells were washed twice with BPS and then lysed in RPPA lyse buffer on ice for 20 minutes, and then were processed by the MDA Anderson's RPPA core facility. The cells were then lysed in TLD buffer with protease inhibitors, where equal amounts of protein were separated, and then blocked in LICOR blocking buffer and incubated overnight with specific antibodies, and then they were washed. The IR dye labeled secondary and antibodies were applied, and then the blots were then visualized with the LICOR Odyssey system, and bands with intensity were quantified. The The cells were then washed again and then quenched with ice-cold methanol. And then the polar fractions were dried and reconstituted. And then metabolic spectra were acquired and then processed in a metabio lab. And then all cellular assays were analyzed by a one-way ANOVA. And they just did some other data testing calculations. Now for the chemical mechanisms. So one of the first chemical mechanisms that happens is BOC protection of the serotonin amine, which basically means that the serotonin hydrochloride reacts with the ditert-butyl bicarbonate in the presence of K2CO3 to form NO-BOC serotonin, which is a nucleophilic substitution reaction. Then SN2 alkylation of phenyl OH happens, where this KCO3 deproteinizes the phenolic acid, phenolic OH, and then phenolic oxide performs SN2 attack on an alkyl bromide and produces a 5-O-alkylated serotonin derivatives. And then for the other, then the BOC group is removed under acidic conditions by, in the presence of HCl, giving the amine chloride salt. This is for the first 9 compounds that they tested. Then for the rest of the compounds, 10 to 13, they started by having a hydroxynol deprotonated form, which forms an indole ion, which reacts with an alkyl bromine through a backside SN2 attack to form a 4-oxygen alkanine ether, which this is through SN2 nucleophilic substitution. Electrophilic addition happens, where the acid protonates the nitroethylene, making it stronger. Then the iodine ring attacks activated nitroethylene and electrophilic aromatic substitution, which produces a leaving group, generating a nitro-substituted side chain. Then the lithium, aluminum, hydrogen 4 reduction of nitro group produces an amine. The NO2 nitro becomes nitroso NO, and then the nitroso becomes a hydroxyl amine NHOH, and then the hydroxylamine becomes a primary amine. Now they took all these compounds that they made and then collected their results, and part of these results can be found in this table, which shows the IC values, which is the half maximal inhibitory concentration of each analog. The table shows that most of the analogues have low IC values, mainly 6 and 10, meaning that they're most effective at reducing the viability of the cells. This leads to the assumption that the structural changes in those analogues lead to stronger anti-cancer activities. They also took 6 and 10 and had a more concentrated study on them, where they produced this graph that shows the plotted values of the concentration of solution against the cell viability for both treated and controlled cells. And as they found that as concentration increased, there was a decrease in viability, meaning there is a cell killing effect. So in their findings, they found that mainly compounds 6 and 10 can kill breast cancer cells. They found that these compounds actively trigger adipose, as shown by the activated caspase 3-7, showing that the cells were undergoing programmed cell death, not just slowing down the growth. They also found that it inhibits mTOR-dependent survival pathways, and it disrupts cell metabolism, allowing the study to reveal the structural changes can greatly impact biological functions. Now, for the real-world application, these findings can be used to create targeted cancer therapy, which can save several lives, as breast cancer is a growing disease that affects thousands of patients every year. It's also a form of repurposing something that's already within the body, such as serotonin, which can lower risks and shorten timelines of discovery. and allows medicinal chemists to work with products that is already known. These findings also contribute to mTOR inhibitor research, combination therapy strategies that already exist, biomarker identifications, and cancer signaling pathway mapping. Anyways, that's my presentation. Thank you for listening.